Indeed, my name is Gestur Vidarsson. I'm from Sanquin Research Institute in Amsterdam, and we are a not-for-profit organization, and we are working in blood transfusion. But we have our own research, which I'm affiliated with, and as such, we are affiliated again with the University of Amsterdam, where our PhD students graduate from. And today, I want to talk to you about. If I can get the next slide. On FcRn, which is a very peculiar IgG receptor. It's an MHC class 1-like molecule associated with the beta-12 microglobulin. And as I said, it's a very peculiar receptor because it's not really expressed on the surface of cells, it's expressed on inside of the cells. And even if it would be expressed on outside of the cells, it wouldn't even bind to IgG, because outside of the cells, pH is regularly neutral, pH 7.4, and actually at those pH, it doesn't really bind to FcRn. It only can do so after being taken up by cells, where it co-localizes in vacuoles or in intracellular compartments that are acidified to a pH of around 6.5, where histidine residues in the Fc part of IgG become protonated, they become positively charged, and only then it can bind to FcRn, which then takes it outside and lets go, basically, and that result being recycling. Or in the case of, for example, placenta cells and other cell types in mucosa, the IgG can also be transported across the cells, and that results in transcytosis. And in this way, we know now that FcRn is responsible for transporting IgG from the gut, and that's how it was initially identified in neonatal rodents. So it's providing immunity, humoral immunity, to the neonatal rodents so taken up from milk, where you have ample IgG in the milk, unlike humans. But in humans, it's the most was originally known to be responsible for transport of IgG from the mother to the fetus, again providing humoral immunity before birth. But now we also know that it's of course, for a very long time now, that it's involved and responsible for the extraordinarily long half-life of IgGs. So providing it with a half-life of 3 weeks instead of, for example, 1 week with similarly sized proteins. It does so also for albumin, and I'm guessing that my colleague Jan Andersen will continue talking about that in more details in the next talk. But it transports also at least IgGs to mucosal surfaces, and also responsible for mucosal immunity. But it's much more complex than that, and this is also what I will end my discussion on, that is also actively involved in phagocytosis and responses on myeloid cells. So these will be my 2 topics. I will start by talking about transport across the placenta and the half-life of antibodies, and some discrepancies that we have been working on and trying to resolve to gain more understanding of the biology of FcRn, and then end my talk on the potential role on myeloid cells. So this is basically what we know of antibodies and FcRn biology. It's responsible for the long half-life of antibodies of 3 weeks. So for IgA, it's like 1 week. So this is true for all the antibody subclasses except IgG3, which has a very short half-life. So it's just like FcRn is not capable of rescuing it, but also placental transport, which is exceptionally good for all subclasses except IgG3, and peculiarly not for IgG2. So it's similar to IgG3, which is a discrepancy on its own, because if FcRn is devoid, cannot rescue IgG3 half-life, so then it's understandable that it cannot also transport across the placenta. But then it's very difficult to understand how it is possible to recycle IgG2 normally, but not transfer across the placenta. So these are obvious discrepancies that deserve clarifications. So these are real life examples of antibody transfer across the placenta. We see this expressed on the y-axis as a ratio of IgG found in the cord blood or in the fetus and from the mother. So the fetus obviously not producing its own antibody. It gets acquired all its antibodies before birth from the mother. And basically what you can see is that it's actively being transported across the placenta because the ratio is above 1. So basically there's higher level of antibodies in the fetus for IgG1 and for IgG4, that's true. But for IgG2 and IgG3, it's lower. So these are basically the first example. And this is something that we've studied actively also together with Andrei, the second speaker of today. And we first started looking at the half-life of recycling of antibodies and looking at agammaglobulinemic patients that don't make IgG on their own, basically allowing us to, and they receive IVIG every other 4 weeks, basically allowing us to study the relative degradation of those subclasses in this human. And basically what we see is that the relative distribution of IgG2, 1 and 4 is more or less not changed. For IgG3, the bulk of IgG3 is disappeared. This is something that we expect. IgG3 has a very short half-life, we knew that. But there is variation in IgG3 because you cannot really say IgG3 is IgG3 because it's very variable. There's a huge number of allotypic variations. So basically meaning that IgG3 differs between individuals quite substantially. Well, most of the IgG3 in Europeans is found with an arginine, so a critical arginine in the FcRn interaction site. This is not true for all IgGs because there is a minor fraction found as individuals in Europe. It's substantially more, and if you go to some of Asian countries and Africa, where you have a substantial proportion of individuals with a histidine at this position, so for position 435. And if we, this allotype is also found in IVIG to a minor degree, but it's still there. And if you measure the relative degradation of this antibody, it's not degraded, at least not compared to IgG1 and IgG3, and definitely not compared to the bulk of IgG3, which has an arginine at this position. So basically in this paper, we basically showed that the reason why IgG3 has a very short half-life is the single amino acid difference between normal IgG1s, IgG2 and IgG4, but IgG3 actually that has a naturally variant of gluconicity has a normal half-life. So basically it's not completely true what it says in the textbook. And also not for transport across the placenta, because indeed, while antibodies of IgG2 and IgG3 has a short or a lessened capacity to be transported across the placenta, IgG3 with this histidine in 4-5 is actually transported to a very similar degree as normal IgG3. So IgG1. So histidine at this position is sufficient to explain this short half-life and short or lower capacity to be transported across the placenta. And actually some individuals, this is not true, some individuals have long half-life of IgG3 and very nice good transport of IgG3. And the question is, why is IgG2 with such a half-life? And I will come back to this later. But I would like to just first have a short intermezzo of why this is important, and especially in the case why we started looking at this, and this is in terms of transplacental transport of antibodies and alloimmunization, where you have paternal antigens on blood cells, for example, platelets, but also red cells, where you have antibody formation against these paternal antigens. And because of FcRn, those antibodies are transported specifically across the placenta, where they cause degradation of these cells, and these can cause life-threatening thrombocytopenias or anemias, depending on the cell type involved. And these antibodies that are transported, they are particularly prone to be IgG1 or IgG3. So it's very important clinically to know if the IgG3 can also be participating in this. But it's also very important to realize that antibody responses against those antigens are very prone to have a very strange fucosylation or glycosylation in the Fc part, where you have lack of fucose. And this causes elevated Fc gamma receptor 3 affinity with elevated ADCC and phagocytosis. So even making these antibodies even more clinically relevant. And we have a series of papers where we described how this and why this is important. But then the question is FcRn involved in somehow in this pathway? And the first is to explain a little bit why this fucosylation is important. So normally antibodies have high levels of fucose, so this glycosylation in the Fc, and this high level of fucose basically instructs the immune system to carry out most of the effective functions, Fc gamma receptor 2 and 3. These are more abundant receptors, at least 2 is very abundant receptor. Fc gamma receptor 1 is less. But once you get an immune response that have lack this fucose, such as in these alloimmune responses in pregnancies, these antibodies have elevated affinities to Fc gamma receptor 3 up to 40-fold, which can translate into black and white, so either absence of ADCC responses to very good ADCC responses, followed by extreme clinical pictures. And the question therefore remains, is FcRn somehow involved? Does it discriminate? Does this pathway discriminate between these glycoprones? So this is basically what we did. We looked at the glycosylation in paired monocard samples, and basically, to make a long story short, we found that there is no change in those glycans. So this is a typical bioengineered glycan plant in the Fc portion. It's very varied with the level of fucose, but even more varied in terms of glycosylation and sialic acid and transfection, basically. If you compare fetal and maternal blood, you see that there is no difference whatsoever in any of the subclasses. And I have to note that we cannot make a distinction between IgG2 and 3 because the glycopeptides are identical. But basically, there is no difference, indicating that at least Fc gamma receptors that discriminate between these glycopromes are not involved in transport, and that FcRn that is most likely only sufficient to transport the antibodies across the placenta does not discriminate between those glycopromes. I think that this is a very nice study by the group of Clark Anderson showing that fetuses in mice of born to heterozygous FcRn mice. So you get either litters that are FcRn knockout or fully FcRn positive or homozygous positive or heterozygous. You see that these pups are born with either absent of antibodies, no antibodies, and either or high levels of antibodies, or even without antibodies. So suggesting that indeed that FcRn is sufficient to explain the transport across the placenta as we expected. And I want to continue a little bit on this pathway because there are ways to intervene in these alloimmune responses, and I guess also in autoimmunity, where you have autoantibody responses against, for example, your own blood cells or your ITP or that's in thrombocytopenia. And the ways how people go about in the clinic and with some experimental things that tap into the FcRn pathway is, of course, the long-standing use of IVIG or high-dose IVIG therapy, or use so basically in IVIG therapies, what you do is that you saturate the FcRn pathway with exogenous antibodies from plasma of other individuals or pool of individuals. You purify the fraction, you give equal amount of antibodies that basically exist in your body, so thereabouts you saturate the FcRn pathway in such a way that the FcRn recycling pathway cannot cope with the amount of antibodies produced to them, and therefore some of the antibodies are being degraded. And that means that the antibodies endogenous of the patient, including autoantibodies, are excessively degraded, and you get alleviation of symptoms. At least that's one way, hypothetically, how IVIG works. And based on this pathway, there are now new therapies that seem to be coming up where you tap into this pathway differentially using anti-FcRn antibodies or antibody fragments. And this is now ongoing in several very promising trials that you can target FcRn directly blocking a recycling of endogenous antibodies, basically doing the same thing in IVIG but done differently. I will show some example of this, and I will then finally end up with another way how to tackle this, and that's by giving an FcRn version of antigen-specific antibodies that will then, in terms of pregnancies, that will be transported across the placenta. You give the antibody to the mother, the antibody is transported to over the placenta. It will find their antigen, the cognate antigen, but it cannot trigger any effector function. So basically, effectively competing with antibodies found in the mother and have effector functions. But that's more theoretically and more experimental. So in IVIG, high IVIG dose therapy, basically an example of how this looks like if you give IVIG. So if you don't give IVIG, you see that the amount of antibodies that you find in the mother basically corresponds with what you find in the cord. So basically, FcRn is not saturated. It seems the more antibodies you find in the mother, the higher levels of antibodies you find in the cord blood. If you saturate this pathway with high-dose IVIG therapy, so basically you exceed the level of normally that you find in individuals with high-dose IVIG, you cannot exceed basically what you find the FcRn pathway cannot cope with this. You get not only less half-life of the antibody in the mother, but also the pathogenic antibodies cannot cross as easily. So basically, in double pathway, you outcompete the endogenous pathogenic antibodies. And this is a very effective therapy, basically in ITP or FNAIT or fetal-neonatal thrombocytopenia and other alloimmune-mediated diseases in pregnancies. Anti-FcRn therapies that are now in clinical practice by several companies, including UCB, Synthion, and Argenx. They have different versions of antibodies targeting. I won't go into that, targeting FcRn. Basically, you see that they very effectively diminish the antibody levels in different subclasses. And I would like to point out that it's very effective for all of the subclasses. Actually, it seems that IgG3 is being degraded even to a further degree quicker, but it rebounds also quite very nicely back. But for IgG2, it seems to be happening more slowly and with a slower desolvation of basically IgG2 seems to rebound very much slower, which is peculiar, and but also is pointing to the fact that I'm suggesting that IgG2 and IgG3 they are different from the other subclasses somehow, which is curious. And then I would like to continue on IgG2 because this is from several of our studies that we initiated basically by a group in Cambridge, the group of Mike Clark, who created an FcRn antibodies for the very reason basically that I suggested to you that you could give an FcRn antibodies to a mother that would be outcompeting pathogenic antibodies in the fetus. And what they did was to make an antibody that contains IgG1 antibody that contains IgG2 determinants and IgG4 determinants in a certain allotypic negative variant. So on its own, it doesn't contain any immunogenetic epitopes, and thereby generating an FcRn IgG1 antibodies that they call delta F antibodies. And then the idea was that these antibodies would be crossing the placenta as effectively as normal antibodies. But actually, in perfused ex vivo perfused human placentas, when we looked at this was actually that did not seem to be the case because wild-type antibodies they were transported efficiently across the placenta, while these delta F antibodies were not. So these are two different backgrounds. So this is an anti-red blood cell, anti-rhesus antibodies, and anti-platelet antibodies, and they did not seem to be crossing the placenta as well as normal antibodies, indicating that there was something wrong with these antibodies. So we took this P2G1 antibody, so G1 stands for IgG1, G2 stands for the same variant, and then IgG2 antibodies. So the delta F antibodies, if you look in transcytosis, so in this kind of system where you give the antibodies in the apical compartment, and you look at transcytosis to the basolateral compartment, you see that in either jar cells, or the procarcinoma, trophic cell-like cells, or melanoma cells that we transfected with FcRn, we basically see that the IgG1 antibodies are transported quite well across that layers of cells, while the skeletal net are deficient in this, so they are transported to a lesser degree with both cell types basically, and to such a degree that it's actually very similar to IgG2, which in essence is not really that peculiar, but it is definitely decreased. So this is particularly interesting in terms that a variant that is basically identical to the delta net, a variant called delta NEC. It's identical except that it contains a glycine 236 that is lacking in the delta NEC variant, and this glycine 236 is part of a dual glycine residues found in all IgG subclasses. It's in the lower hinge, basically here, actually belonging to the CH2 domain. And all the subclasses and all species, or most species, they contain these two glycines, except human IgG2, where it only has one glycine. So this delta NEC lacks this glycine, has a very similar transport to an IgG2, but if you actually have the same variant and you with the glycine, it's transported equally to an IgG1 variant, and this is in both cell types. Because these antibody variants has a lot of different other mutations, we decided we have to confirm this. We make a new antibody variant, antipneumococcal antibody. That's not important, but basically what we see is that for IgG1 wild type and IgG2, we see the same. That IgG2 are transported less well across this placenta. If you now delete glycine from IgG1, you get a reduced transport of this antibody to an equal level actually of wild type IgG2. If you do the reverse experiment, you take a wild type IgG2 and you put glycine 236 in there, so making it more IgG1-like, you actually get transport back to human IgG1 transport. So this is transport, and this is actually reflected also in what we see and collect at 24 hours. We see that the total amount of antibodies that we find after 24 hours is reduced for the glycine, the variants of antibodies that have only one glycine in their hinge. So that's wild type IgG2 and an IgG variant where we deliberately remove the glycine 226. But it's equal to an IgG1 for IgG2 variant where we add this glycine 226. So basically indicating that somehow glycine 226 is interfering with this pathway of FcRn transport, and this is very difficult to understand because, as I mentioned, the FcRn interactions are in the CH3 domain of the antibody. So this is the Fc domain. These are the fat fragments. Here are the key residues involved in binding, so the histidine residues involved in binding to FcRn, and the two glycine residues are basically found here in the lower hinge region, which is obviously not close to this binding interface. The question is, why is this? How is this affecting? How can this affect FcRn transport? And of course, the actual reason is unknown, but it's tempting to speculate that it has something to do with the stoichiometry of this complex, because obviously FcRn cannot bind to an antibody in the standing up position because it will clash with the lipid membrane. Somehow something has to give, and probably FcRn needs to bend back towards the membrane, and probably an antibody molecule also needs to bend the fat fragments away from the fragment, from the bilipid membrane. Because obviously otherwise this complex cannot exist. And it's even more interesting, I think, in terms that we know now that charged residues in the fat region can strongly affect half-life of antibodies, so also far away from FcRn binding site. And I think that seems to also indicate that the composition and the stoichiometry of this complex is important. And it's even more interesting in terms of data coming from old data from the lab of Pamela Bergman and Sally Ward, indicating that the transport vesicles of FcRn and antibodies is not simple vesicles, but actually tubular structures where you have FcRn on bilateral membranes, where you have two FcRn molecules that are actually required for full efficient transport across the membrane. So basically you get much more close proximity here. So flexibility of both FcRn and the antibody is probably important. I would like to switch gear, as I promised, to talk a little bit about the presence of FcRn on the myeloid compartment. Besides, we and others have found that FcRn is actually quite highly expressed on the myeloid compartments, not on T cells or NK cells, or at least not highly expressed there, but it's particularly highly expressed on neutrophils and monocytes or phagocytic cells. And I think it's very nice data coming from the Jackson laboratories, where they actually showed that the contribution of the myeloid compartments in providing the long half-life of antibodies is actually very important, because if you have, and where they did actually experiment, a bone marrow transport experiments in using either knockout bone marrow, FcRn knockout bone marrow, transporting that into knockout or wild-type mice, or FcRn sufficient bone marrow, where they could look at the contribution of either compartment. And basically, as we know, half-life of antibodies is very long if you have FcRn in all cells of your body, basically, that do normally express it. If you lack FcRn in both compartments, it's very short. But if you have only FcRn in either the myeloid compartment or the rest of the compartment, you see sort of that the whole system seems to meet in the middle, suggesting that the half-life of antibodies is not, it seems to be even up to 50% being contributed by the myeloid compartment. But we think it's a little bit more complex than that. It's not only half-life, because as we described quite some time ago, that FcRn also seems to tap into the phagocytic process itself, because if you lack FcRn, phagocytosis is much less. And what we think is happening is basically that FcRn is residing in granules within the cells. And when you get triggering through Fc gamma receptor on the surface, you get degranulation of these cells, or of these vesicles. You get the engulfing phagosome or phagocytic synapse or immunological synapse that is somewhat close compartment due to integrine interactions through it, depending on the target, how that works. But at least protons are pumped into this compartment where FcRn start to be, at some point, being able to bind and participate in the engulfment, complete ingestion of those antibody targets. And this is perhaps even more interesting in the light of that monocytes also express this. Monocytes that can be transformed and mature into dendritic cells that take up antigens, and in the states where they are very capable of doing this, into states of dendritic cells, FcRn expression goes dramatically up, and it goes back down again in the mature states of dendritic cells. And basically, work of Sally Ward and in particular the Bloomberg's lab seem to suggest that there is a handing over mechanism where triggering through Fc gamma receptor leads to engulfment of immune complexes, where FcRn takes over when it becomes acidified, transport this immune complex further downstream, where this antigen broken down and presented on energy class 1 and energy class 2 molecules for antigen presentation. And basically, a very nice work of Bloomberg Lab and Sally Ward have shown this, and I think this is one of the first papers showing this, basically, that if you give, you challenge, or you give immunized mice with antigens, and you challenge T cells that after that with immune complexes through dendritic cells, taking up these immune complexes, you look at proliferations of T cells that you need FcRn to do that, because if you have FcRn knockout dendritic cells, they cannot provide this proliferation or antigen presentation to those T cells. Or if you do it in wild-type dendritic cells with immune complexes consisting of antibodies, you cannot find FcRn. They also flatline and are not capable of providing antigen presentation. And this has been nicely confirmed in various papers, except one paper from our lab that actually, together with the group of Grada van Bleek in the Utrecht University, where we actually looked at different kind of antigen with the synthetic virus or a virus challenge in mice, where we give it intranasally. And there we've actually found no traces of that FcRn capable of providing stimulus to dendritic cells to T cells. They're not providing any extra proliferations of these cells. While if you knock out Fc gamma receptor, then this boost that you get by using antibodies. So antibodies do provide an immune boost and proliferations of the immune cells, T cells. But if you knock out the Fc gamma receptor, this gain is lost. And basically, FcRn, we also are not capable of providing this with antibodies to artificial antigen TMP, and there is no deficient proliferation using these antibodies that either lack FcRn binding. And further looking into this, we were not capable of looking into different subsets of see no sufficient in this model of dendritic cells that lack FcRn in the system providing. So you see immune enhancement by antibodies of immune sera that, or using palivizumab in this case, of different T cell populations, but there is no sufficient enhancement of this response if you use dendritic cells that lack FcRn expression. And even if you go and look into the humoral immune response, we did not see any sufficient responses. Perhaps 4 weeks after priming, we see somewhat lower titers of antibodies in these mice. But again, it's very difficult, of course, to tease apart the half-life effect towards from the antigen presentation pathway here. But at least 6 days after challenge, we see definitely no change in antibody titer in these mice. And the question is why? Is that I think one of the major differences that other labs have provided, they always use soluble protein antigen as starters, while we use membrane embedded antigens from RSV that has been UV treated, and we don't know exactly how that affects the system. So perhaps this is a differential way how antigens evolve, tap into this pathway. But I think the majority of the work seems to suggest that this is true, that there is a handing over, there is a collaboration between these two pathways, that there is a phagocytic that you require the Fc gamma receptors, but also later on you require FcRn to take up and process these antigens and to send them to T cells. But I think that's even more naive. It's much more complex than that, because there has been a number of studies actually showing that in different settings that you can actually tolerize mice through this pathway, and especially in this kind of settings where you challenge mothers with antigens, where you get that can in these mice lead to anaphylactic responses. But if you have mice that are born to these mice, that the neonatal offsprings, that they take up this antigen through the feeding breast milk, and basically that later in life that they are sensitized, they are not reacting with strong anaphylactic responses. So basically, FcRn is basically a very complex biology, because at least in this case, the antigen processing can lead to boosting. It can either do nothing, or it can lead to tolerization, it seems. So it's much more complex than we initially thought, where we thought it was only recycling IgG or transporting across tissues like the placenta and mucosal tissues. It's of course also very important still requires distribution of antibodies, and all antibody drugs need to go, I guess, through this engineering stages where you can tap into this pathway and try to boost them in a way that are beneficial for future drugs. I think it's also important to realize indeed that FcRn is much more, it's not only that, it's also important in different processes, particularly in myeloid cells. And with that, I would like to thank my collaborators. I think the PhD students that at different stages in this long process have been involved. Our collaborators in Utrecht who did the RSV work, Jan Terje with IgG3 work, and our colleagues in Cambridge on the FcRn antibodies and Manfred Wurer for the glycosylation analysis. And with that, I would like to thank you, and I would be welcome to take any questions later on.